Today, we are going to discuss about stability of protein structure. Proteins are large biomolecules composed of different amino acids which form its basic unit. Protein structure is important for its function, dictating how it folds into specific shapes and interacts with other molecules. Then maintaining the stability of protein is important for the functioning and biological activity. Protein stability is a topic of major interest for the biotechnology, pharmaceutical, and food industries. In addition to being a daily consideration for academic researchers studying proteins, proteins are complex molecules with complex three-dimensional structures that determine their specific roles within cells. These structures are susceptible to disruption by various factors like changes in temperature, pH, or exposure to chemicals. When a protein loses its native structure, it is said to be denatured, making it non-functional. And this can lead to cellular malfunctions and potentially contribute to different diseases. So, stability is the potential of a pattern to survive over time and therefore is integral to our understanding of biological systems and their evolution processes, such as protein unfolding, denaturation, degradation, conformational changes, then enzymatic modifications, and proteolytic cleavage may transfer this pattern. Protein stability. Protein stability is the net balance of forces which determine whether a protein will be in its native folded conformation or a denatured state. Protein stability may be compositional stability or conformational stability. Compositional stability means that a protein molecules must have the same chemical makeup. The chemical homogeneity of a sample can often be determined using mass spectrometry or an SDS-based gel. Compositional homogeneity is typically compromised by post-translational modifications like glycosylation and proteolysis, which can affect the primary structure of the protein molecules and generate compositional variability. Second is conformational stability. Conformation means shape of a protein. A large class of proteins referred to as intrinsically disordered proteins, IDPs, contain significant levels of conformational disorder and in some cases have no discernible three-dimensional structure at all. It is estimated that 40% of all human proteins contain at least one disordered segment and 25% are completely uh, disordered. So protein stability means different things to different people. A pharmacologist, a biotechnologist, or food scientist may primarily consider the half-life of a protein's uh, activity as a measure of its stability. However, a protein chemist or a structural biologist may concern themselves with changes in the primary, secondary, tertiary, or quaternary structure of a protein as a measure to its stability, right? Then stability of the protein on a structural level. So first we'll start with the primary structure. Primary structure of a protein, uh, just a sequence of amino acid. Now you can see here, this is sequence of amino acid. So this is a primary structure of a protein. Sequence of amino acids in the polypeptide chain can be modified in several ways by post-translational modifications. Post-translational modifications are the result of many different changes to the primary structure of a protein, including proteolytic processing, protein splicing, and the addition of other functional groups to the amino acids. Post-translational modifications can affect both compositional stability, as the modifications may be non-uniform or incomplete, and also conformational stability, as the modifications may affect protein disorder and dynamic. This is illustrated by glycoproteins, which are often not uniformly glycosylated at all possible glycosylation sites, therefore leading to compositional heterogeneity. Next is secondary structure. Protein secondary structure is a localized three-dimensional structure of a polypeptide chain. You can see here secondary structure. Secondary structure can be described in terms of pattern of hydrogen bonding between amide hydrogen atom, this one, and carbonyl O atoms of the backbone. You can see here. Helix is the predominant type of secondary structure, accounting for approximately one-third of all secondary structure elements. The stability of the folded protein is largely dictated by amino acid composition, and as such, the primary structure results in a unique kinetics minimum of free energy. Uh, on a simplified level, the primary driving forces behind the formation of secondary structure and in turn tertiary structure are hydrogen bonding and hydrophobic interactions, right? Next is tertiary structure. The tertiary structure of a protein is the overall shape or fold adopted by the polypeptide chain. Many factors affect the process of protein folding. You can see here, this is tertiary structure of a protein. This structure means here many factors affect the process of protein folding in tertiary structure, including conformational and compositional stability first, then cellular environment, including temperature and pH, then primary secondary structure, mainly bondings, hydrogen bondings, right? Salt bridges, hydrophobic effects, van der Waals forces, ligand binding, cofactor binding, ion binding, post-translational modifications, right? So what are hydrophobic interaction? You can see here. Here, these are hydrogen bonds, disulfide bridges, hydrophobic interactions, hydrophilic interactions, ionic bonds, right? So all these type of the bonds are responsible for formation of tertiary structure of a protein. Hydrophobic interaction means here these amino acids orient themselves toward the center of the polypeptide to avoid the water, right? Then disulfide bridges, you can see here. Disulfide bridges, here amino acid cysteine forms a bond with another cysteine through its R group. Then hydrogen bonds, you can see here hydrogen bonds. Here polar R groups on the amino acids form bonds with other polar R group, right? Then hydrophilic interactions. Here these amino acids orient themselves outward, right? Outward to be close to the water. Then ionic bonds. You see here. Here positively charged R groups bond together, right? Next is quaternary structure. Quaternary structure is the arrangement of the folded protein subunits into a multi-subunit complex, as you can see here, right? Multi-subunit complex. These different subunits like this here you can see four subunit. So quaternary structure is also stabilized by non-covalent interactions and disulfide bonds, as in the tertiary structure, where more than one polypeptide is held together to form a single functional unit called multimer, right? This is a multimer. The stability of these complexes is of importance for the regulation of allostery and cooperatively, which often results from the conformational changes within individual polypeptide chains. Next is the stability of proteins is determined by various forces, right? Means different type of the forces responsible for the stability of protein structure. First is hydrophobic effect. Already now we have discussed hydrophobic effect. Here, primary driving force behind protein folding is the hydrophobic effect, which refers to the tendency of hydrophobic molecules to avoid contact with water, as I already told you. Then, hydrogen bonds, right? Hydrogen bonds form when hydrogen atom bonded to an electronegative acceptor atom interact with another electronegative acceptor atom carrying a lone pair of electrons. Disulfide bonds. Disulfide link provide proteins folded states a great deal more stability, as in many situations simply reducing the cysteine connections is enough to cause unfolding. Instead of being enthalpy, the source of stability seems to be entropic here. By restricting the degrees of freedom that the disordered polypeptide chain can exercise, the addition of disulfide bond lowers the entropy of the unfolded state. Then ionic interactions. Ionic interactions, uh, these interactions typically have minimal impact on protein stability due to strong solvation of isolated ionic groups by water. So salt bridge, also known as ion pair, occur when two ionic groups with opposing charges interact within a protein, means a feature found in most proteins. Dipole-dipole interactions. Dipole-dipole interactions which result from the close association of induced or permanent dipoles are weak. Otherwise, these are weak interactions. Van der Waals interactions are the name given to these forces. Right? Why protein stability is important? Stable protein maintain their native conformation, preserving their biological activity during manufacturing, storage, and administration. Second, providing an understanding of the basic thermodynamics of the process of folding. Then increased protein stability may be of value in food and drug processing and in biotechnology and protein drugs. Then in vitro stable protein enable reliable assays and high throughput screening, while in vivo they ensure proper pharmacokinetics and therapeutic outcomes. Then factors affecting protein stability, pH. Here, pH in general electrostatic interaction are believed to contribute to a small amount of the stability of the native state. However, there may be exceptions. So pH here proteins are most stable in vicinity of their isoelectric point. Isoelectric point of a protein is defined as the pH at which the net charge of the protein molecule is zero. Proteins are positively charged at a pH below their pi and negatively charged at the pH above their pi. Then ligand binding. It has been known for a long time that binding ligands are, for instance, inhibitors to enzymes, increase the stability of the proteins. Then disulfide bonds. For many proteins, if their disulfides are broken, then carboxymethylated with iodoacetate, the resulting protein is denatured. Then temperature. High temperature increase kinetic energy, which cause molecules to vibrate rapidly and forcefully. This result in the disruption of hydrogen bonds and other nonpolar hydrophobic interactions, leading to denaturation of the protein. Then agitating, like shaking, vortexing, and stirring the solution vigorously can upset protein stability, right? Then methods to assess stability of proteins, different methods like thermodynamic approaches, then spectroscopic techniques, structural methods, computational simulations. Thermodynamic approaches involve measuring change in free energy associated with protein folding and unfolding. Spectroscopic techniques like fluorescence spectroscopy. Thanks for watching. If you like this video, make sure to subscribe for more. Thank you.